Hello my dear friends, welcome back to CSIR at home. In this video, we can discuss the important topic for CSIR examination. The important topics are glycolysis and its regulation. Okay, regulation is important. Then electron transport chain and its inhibitors. Then protein sorting, in which N-linked protein glycolization is very important. Okay, then transcription, cancer, ABC model of flowering, that is very important. Then phytochromes, oxygen saturation curve, pedigree analysis, co-evolution, Hardy-Weinberg equation, hybridoma technology, restriction enzymes, and RNA silencing. These are the important topics for CSIR examination. Now we can discuss the timetable. Day 8. Morning 5 to 6 a.m. study glycolysis. We have videos on glycolysis. Just watch it. Then 6 to 7 a.m. on-go genes. You can refer CAR page number 657. Then 9 to 10 a.m. study co-translational and post-translation protein translocation. Bruce Alberts. 10 to 11 a.m. study transcription basics. Which book to refer? Lodish. Then 11 to 12 p.m. study tumor suppressor genes. Then 12 to 1 p.m. study ABC model of flowering. Which book to refer? Segrantis. Then 3 to 4 p.m. study structure, function and mechanism of action of phytochromes. Segrantis. Okay. Then 4 to 5 p.m. study oxygen hemoglobin dissociation curve. Tortora. 6 to 7 p.m. study pedigree analysis, symbols. This is very important, pedigree analysis. Then 7 to 8 p.m. study insect-plant co-evolution. Which book to refer? Mark Ridley. Then 9 to 10 p.m. study Hardy-Weinberg equation, Peter Stilling. Then 10 to 11 p.m. hybridoma technology, BDSN. Day 9. Morning 5 to 6 a.m. study glycolysis regulation, STRIR. 6 to 7 a.m. study virus-induced cancer, Bruce Alberts. 9 to 10 a.m. study signal hypothesis, Bruce Alberts. Then 10 to 11 a.m. the phases of transcription, Watson. Then 11 to 12 p.m. study transcription in bacteria, Watson. Then 12 to 1 p.m. ABC model of flowering, Segrantis. Then 3 to 4 p.m. phytochromes. Then 4 to 5 p.m. oxygen hemoglobin dissociation curve, okay, Tortora. Then 6 to 7 p.m. pedigree analysis, Benjamin. Then 7 to 8 p.m. parasite host co-evolution. Then 9 to 10 p.m. Hardy-Weinberg equation. Then 10 to 11 p.m. hybridoma technology. Day 10. Morning 5 to 6 a.m. 5 to 6 a.m. solve previous year questions on glycolysis. Then 6 to 7 a.m. metastasis. Then 9 to 10 a.m. study how endoplasmic reticulum signal sequences and SRP direct ribosomes to the endoplasmic membrane. Which book to refer? Bruce Alberts. 10 to 11 a.m. N-linked protein glycolization in the rough endoplasmic reticulum, Bruce Alberts. Then 11 to 12 p.m. RNA polymerase structure. Then 12 to 1 p.m. ABC model of flowering. Then 3 to 4 p.m. phytochromes. 4 to 5 p.m. oxygen saturation curve. Okay. The oxygen saturation curve of hemoglobin in patients with hexokinase and pyruvate kinase deficiency, void and void. Then 6 to 7 p.m. autosomal traits, pedigree analysis. Then 7 to 8 p.m. revise photosynthesis, okay? Revise photosynthesis. 9 to 10 p.m. restriction enzymes, T.A. Brown. 10 to 11 p.m. previous year questions on part A. Day 11, morning 5 to 6 a.m. study glycolysis regulation. Which book to refer? STRIR. Then 6 to 7 a.m. angiogenesis. Okay, 6 to 7 a.m. angiogenesis in cancer cells. Then 9 to 10 a.m. 9 to 10 a.m. protein translocation. 10 to 11 a.m. synthesis of the N-linked precursor oligosaccharide in the rough endoplasmic reticular membrane. Then 11 to 12 p.m. transcription termination, Watson. Then 12 to 1 p.m. revised replication. 3 to 4 p.m. phytochromes, okay, Segrantis. Then 4 to 5 p.m. work on the questions, okay, on part A questions. 6 to 7 a.m. pedigree analysis. 7 to 8 p.m. pedigree analysis. Then 9 to 10 p.m. the recognition sequence for some of the most frequently used restriction endonuclease. This is very important, okay? Restriction endonuclease. Then 10 to 11 p.m. RNA silencing primrose. Then day 12 electron transport chain components. Which book to refer? STRIAR, page number 530. Then 6 to 7 a.m. electron transport chain Q cycle. That is also very important. 9 to 10 a.m. the integration of a double-pass transmembrane protein with an internal signal sequence into the endoplasmic reticular membrane. 10 to 11 a.m. the role of N-linked glycosylation in endoplasmic reticulum protein folding. Okay. 11 to 12 p.m. transcription in eukaryotes. Then 12 to 1 p.m. transcription factors. Which book to refer? Watson. Then 3 to 4 p.m. phytochromes. 4 to 5 p.m. revised cell signaling. Then 6 to 7 p.m. pedigree analysis problems. Okay. Then 7 to 8 p.m. part A questions. 9 to 10 p.m. how to perform a restriction digest in the laboratory. Which book to refer? T.A. Brown. Then 10 to 11 p.m. RNA silencing primrose. Day 30. Morning 5 to 6 a.m. cytochrome C oxidase mechanism. Which book to refer? STRIAR. 6 to 7 a.m. structure of ATP synthase, STRIAR. Okay. Then 9 to 10 a.m. inhibition of electron transport chain. Then 10 to 11 p.m. the export and degradation of misfolded endoplasmic reticular proteins. Okay. Then 11 to 12 p.m. the unfolded protein response, Bruce Alberts. Then 12 to 1 p.m. spliceosome mediated splicing reaction, Watson. 3 to 4 p.m. revised ABC model. Then 4 to 5 p.m. part A questions. Then 6 to 7 p.m. revised blood clotting cascade. Then 7 to 8 p.m. revised development biology signaling. Okay. Then 9 to 10 p.m. mapping the position of different restriction site in a DNA molecule. Then 10 to 11 p.m. RNA silencing, primrose. Day 14. Revise. You can revise. Okay. You can revise everything you have studied in this week. Okay. Therefore, you can revise glycolysis, then you can revise cancer and cell cycle, then revise electron transport chain, then revise protein sorting, then you can study about transcription group 1 and group 2 introns, Watson. Then alternative splicing, Watson. Then you can revise transcription, phytochromes, methods, and just do some problems on pedigree analysis, then revise oxygen hemoglobin dissociation curve. Okay, this is about the timetable. Okay, if you like this video, please like, share and subscribe. Thank you.